In mammals, the egg is protected by a layer of extracellular matrix consisting mainly of glycoproteins called the zona pellucida. When a sperm binds to the zona pellucida, a series of biochemical events called the acrosomal reactions take place. In placental mammals, the acrosome contains digestive enzymes that initiate the degradation of the glycoprotein matrix, protecting the egg and allowing the sperm plasma membrane to fuse with the egg plasma membrane. The fusion of these two membranes creates an opening through which the sperm nucleus is transferred to the ovum. Fusion between the oocyte plasma membrane and sperm follows and allows the sperm nucleus, centriole, and flagellum, but not the mitochondria, to enter the oocyte. The nuclear membranes of the egg and sperm break down and the two haploid genomes condense to form a diploid genome. This process ultimately leads to the formation of a diploid cell called a zygote. The zygote divides to form a blastocyst and upon entering the uterus, implants in the endometrium beginning pregnancy. The development of a multicellular organism begins from a single-celled zygote, which undergoes rapid cell division to form the blastula. The rapid multiple rounds of cell division are termed cleavage. After the cleavage has produced over 100 cells, the embryo is called a blastula. The blastula is usually a spherical layer of cells, which is called the blastoderm, surrounding a fluid-filled or yolk-filled cavity, which is called blastocell. Mammals at this stage form a structure called the blastocyst, characterized by an inner cell mass that is distinct from the surrounding blastula. During cleavage, the cells divide without an increase in mass. That is, one large single-cell zygote divides into multiple smaller cells. Each cell within the blastula is called a blastomere. Cleavage can take place in two ways: holoblastic, or total cleavage, or meroblastic, partial cleavage. The type of cleavage depends on the amount of yolk in the eggs. In placental mammals, including us humans, where nourishment is provided by the mother's body, the eggs have a very small amount of yolk and undergo holoblastic cleavage. Other species such as birds, with a lot of yolk in the eggs, to nourish the embryo during development, undergo meroblastic cleavage. In mammals, the blastula forms the blastocyst in the next stage of development. Here, the cells in the blastula arrange themselves in two layers, the inner cell mass and an outer layer called the trophoblast. The inner cell mass is also known as the embryoblast. This mass of cells will go on to form the embryo. At this stage of development, the inner cell mass consists of embryonic stem cells that will differentiate into the different cell types needed by the organism. The trophoblast will contribute to the placenta and nourish the embryo.
So after the implantation of the blastocyst in the endometrium of the uterus, another process begins, which is called the gastrulation. So to define gastrulation, it is the early phase of the embryonical development in which three germinal layers are formed, namely the ectoderm, the mesoderm, and the endoderm. I will explain later how the cells of every germ layer eventually form specific organs, systems, and structures found within a human body. So let us begin with the ectoderm. The ectoderm is the external layer of the developing embryo. It includes the integumentary system of the human body. Integumentary meaning the outer part of the skin. So it includes the skin, the nails, and the teeth. So because of the invagination process, which I will show later, a portion of the ectoderm will make its way into the developing embryo, and this will form the nervous system of the human body, which includes the brain and the spinal cord. I would also like to add that it also includes the nerve cells that are part of the peripheral nervous system. So it also forms the pituitary gland, which is under the hypothalamus of the brain. So let us discuss the innermost layer, which is the endoderm. The endoderm layer forms the epithelial layers of our lungs, our digestive system, the respiratory system, pancreas, and the liver. It also forms the thyroid and the parathyroid glands. Finally, the mesoderm, or the middle layer of the developing embryo, which is found between the layers ectoderm and the endoderm. So it makes sense that the mesoderm basically makes up everything between the skin and the digestive epithelium. It forms the musculoskeletal system that includes the bone and the cartilage. The three types of muscles, cardiac, smooth, and skeletal. Since it forms the cardiac muscle, it also forms the heart as well as the blood vessels. It also forms the excretory and the reproductive system. So, these are the three layers that are formed during the process of gastrulation. So, the question is, how does gastrulation actually take place? I will explain the process as to how these germ layers are formed. Let us begin with the first diagram. Let's recall what's implantation. Yung implantation, nangyayari siya kapag ang blastocyst, ini-implant niya yung sarili niya sa endometrium, yung lining ng uterus. The blastocyst contains of a region known as the trophoblast. Here is the inner cell mass. Itong inner cavity naman na to, it contains a fluid that provides nutrition to the developing cell. It is called the blastocele. Itong inner cell mass, it will eventually form three distinct germ layers. While the cells in trophoblast, ito yung magpo-form ng chorion as well as the placenta, which are structures that provide nutrition to the developing fetus. Itong trophoblast, nag-e-excrete siya ng digestive enzyme. Ito yung mag-aalaw sa blastocyst to make its way entirely into the endometrium. May kita dito sa diagram na to na yung blastocyst na entirely nasa endometrium na siya. Itong inner cell mass, may kita na differentiated na siya as to different types of cells. Yung hypoblast, pati epiblast. Yung hypoblast cells, eventually, siya yung magpo-form ng endoderm, while yung epiblast, siya yung magpo-form ng ectoderm. Para sa mesoderm naman, magpo-form siya sa gitna ng dalawang layers na to, through a process called evangination, which I will show later. Yung upper portion nitong hypoblast, pati yung upper portion nitong epiblast, it will eventually migrate upwards towards this pole, as you can see here. Yung hypoblast, it developed into the umbilical cord vesicle, which is called the yolk sac sa non-humans. Yung umbilical vesicle, it eventually became part of the umbilical cord system. Yung amniotic cavity naman, na-form siya galing sa epiblast. Dito magpo-form yung organism. We also have this extension. Ito yung tinatawag na chorion, which will eventually be the placenta. Makikita naman dito sa diagram na to, yung mesoderm na nag-form sa gitna ng amniotic cavity pati ng umbilical vesicle. What happens is, if we zoom in here, ito yung tinatawag nating evangination process, wherein the blue layer, nag-e-evanginate siya, meaning, nagmo-move siya inwards. So, as they move inwards, yung blue cells na yun, nagde-develop siya into these red cells. Ito yung tinatawag nating mesoderm. And as the pushing process takes place, the red cells are pushed around the entire structure, which I will show later. Yun yung umbilical vesicle, pati yung amniotic cavity, dun magpo-form yung organism. So, dito naman, sa diagram na to, ayan, may kita na yung mesoderm. Dahil dun sa pushing process, push siya around sa entire structure. Yan. So, the next, and so for the last diagram, makikita dito yung itong entire structure na to. Ito yung tinatawag natin na developing embryo. Yung red portion, siya yung actual embryo, along with the light blue portion and the blue portion. Itong red, di ba siya yung mesoderm natin? So siya yung, basically, siya yung magpo-form everything found in between the light blue and the blue layer. Yung blue layer or yung ectoderm, di ba siya yung external layer? Siya yung magpo-form ng skin, ng ears, ng nails, ng hair, etc. Notice yung blue layer na to, eventually, di ba, mag-move ulit siya inwards. Itong blue layer na to, mag-move ulit siya dito. Dahil nga dun sa evangination process. So, it will form the nervous system. Yung nervous system consisted siya ng brain pati ng spinal cord. Yung light blue layer naman, itong light blue layer, siya yung magpo-form ng digestive system natin. Siya yung magpo-form ng epithelial layer ng digestive system, ng liver, ng pancreas, etc. The outside portion, itong outside portion na to, yan yung tinatawag nating chorion. Tapos yung extension na to, yun yung placenta. Yung structure here, ito yung nagko-connect ng placenta pati ng chorion. Ito yung embryonic stalk na tinatawag natin. Yung amniotic cavity, ito yung nagko-contain pati nag-house sa lumalaking fetus. There, so that concludes the whole process of gastrulation.
As a summary, the three germ layers that are formed during the process of gastrulation are the following: the ectoderm, the mesoderm, and the endoderm. So as I mentioned earlier, the ectoderm is the outer layer of the embryo. It basically forms the epidermis of skin and its derivatives, the nervous and sensory systems, pituitary gland, jaws and teeth, and the germ cells. Secondly, the mesoderm, which is the middle layer of embryo. It basically forms the skeletal and muscular systems, the circulatory and lymphatic systems, excretory, reproductive systems, dermis of skin, and adrenal cortex. Lastly, the endoderm is the inner layer of the embryo. It basically forms the epithelial lining of the digestive tract, the epithelial lining of respiratory, excretory, and reproductive tracts and ducts, and lastly, the thymus, thyroid, and parathyroid glands.
So I'll be giving you an example for the gastrulation of a chick embryo. Here is a video presentation in order for you to understand better. For organogenesis, it is the development of the three layers into organs. Its difference from the gastrulation is that this is more of the development of the three layers, which are ectoderm, endoderm, and mesoderm, into organs, while for gastrulation, it is more of the formation of these three. So under organogenesis, there are four terms to be defined to you. First, what is neurulation? Neurulation forms hollow dorsal nerve cord, or it is simply the formation of the neural plate. What is neural plate? Neural plate is the primary composition that forms the neural tube and to be further developed into brain and spinal cord. So notochord is a stiff dorsal skeletal rod and is formed from mesoderm. It induces the formation of the neural plate and it sends signal for the organs to be formed properly and appropriately. Lastly, somites form blocks of mesoderms arranged along the notochord. It is the sign of segmentation or simply partitions. For the early human embryonic development, I would be discussing to you from the part where the blastocyst reaches the nucleus until it has produced a three-layered embryo with 4 extraembryonic membranes. For the first phase, the blastocyst reaches the uterus. It is pretty explicitly given, so I don't have to elaborate further. For the second phase, the blastocyst implants itself into the uterus. This happens 7 days after the fertilization, and it is also in this part where the expansion of the trophoblast occurs as it implants itself into the uterus. For the third phase, the extraembryonic membrane starts to form at 10 to 11 days after the fertilization. The yolk sac begins to form from hypoblast, extraembryonic mesoderm cells forms from epiblast, and the chorion forms from trophoblast. Gastrulation also begins in these phases, and it happens 13 days after fertilization. Lastly, for the fourth phase, gastrulation has produced a 3-layered embryo, which is ectoderm, mesoderm, and endoderm, with 4 extraembryonic membranes, as stated earlier, which are amnion, chorion, yolk sac, and allantois. And that is for the early human embryonic development.
In gastrulation, it is a ball of cell with a fluid cavity. The cavity is the blastocell, while the embryo is currently at the blastocell stage of development. A blastocele contains a large yolk-filled cell at the vegetal pole, while smaller cells are located at the animal pole. There are 3 colors, and each color represents each tissues. Yellow indicates endoderm, red indicates mesoderm, and blue indicates ectoderm. At the beginning of gastrulation, a few surface cells, called bottle cells, moves into the interior of the embryo, followed by other surface cells. We can track its movement if we would add a dye to a few surface cells. The movement of the embryo creates a lip. It is called the dorsal lip, wherein cells continue to move into at the same time at the ectoderm. While the ectoderm stands in the process called epiboly. As gastrulation proceeds, a cavity called archeteron forms while the blastocell progressively shrinks at the archeteron. Is the primitive guide, which is completely surrounded by ectodermal tissues. Amniotic embryos for reptiles, birds, and mammals. It is a development in fluid-filled sac within a shell or uterus, wherein amnions fluid protect embryos to prevent dehydration or the cushion's mechanical shock. There is also a yolk wherein it stores the nutrients in eggs. Mammalian eggs also store little food. For the patterns of development, we have cytoplasmic determinants, wherein chemical signal such as mRNAs and transcription factors influence pattern of cleavage. Induction. It is where the interaction among cells that influences their fate cause change in gene expression. Totipotent cells is capable of developing into all different cell types. Also, all cells of mammalian embryos and totipotent until the cell 16 stage.